Okay, let's dive right into this. Welcome to this explainer, where we're going to explore what is arguably one of the most incredible frontiers in all of modern science. I want you to imagine, just for a second, that the entirety of human DNA, the actual biological code that makes you, you, is essentially a vast, incredibly complex instruction manual. Now, for the longest time in human history, this manual was completely closed off to us. We couldn't read it, let alone understand what it was saying. But today, well, today not only have we figured out how to crack open that book and read the letters inside, but we're actively learning how to pick up a pen and rewrite the text ourselves. It's an absolutely profound shift in how we interact with biology. And today, we're going to break down exactly how scientists pull off this incredible feat. So here is our roadmap for the explainer today. First, we'll look at reading the genomic manual. Second, accuracy and sequence trade-offs. Third, cutting and rewriting DNA. Fourth, CRISPR and editing tools. And finally, we'll wrap up with the future of precision medicine. All right, let's get started with Section 1, Reading the Genomic Manual. To really grasp this, think of genetic sequencing less like sitting down to read a novel straight through, and more like being handed a billion-piece jigsaw puzzle that you have to piece together to reveal the final picture. To read the genome, scientists follow 4 core steps. First up is library preparation. Because the DNA molecule is simply way too massive to read in one go, genomic DNA is intentionally fragmented. It's broken down into tiny pieces to build what we call a genomic library. Next is massively parallel sequencing. And this is where the magic of modern tech really shines. Instead of reading one piece at a time, machines read millions of these sequence fragments all at once, which produces millions of short sequence reads. Step 3 is assembly. Now that we have this massive pile of read fragments, computers take them and align them against a known reference genome. They're essentially using the picture on the front of the puzzle box to figure out where all the tiny pieces belong. And finally, step 4 is variant calling, where we look at our newly assembled puzzle and pinpoint exactly where it differs from that standard reference picture. Now, when we do that variant calling, what we're really hunting for are single nucleotide variants, or SNVs. If the genomic manual has 3 billion letters, think of an SNV as a single typo. Sometimes that typo does absolutely nothing at all, but other times that one specific difference, identified when our assembled sequence reads are aligned against the reference genome, is the exact root cause of a severe genetic condition. Finding these SNVs is basically the whole point of reading the manual in the first place. Moving right along to Section 2, accuracy and sequence trade-offs. Look, as incredible as the sequencing process is, the reality of reading 3 billion letters of microscopic code is pretty fraught with practical challenges. We simply can't read every single letter perfectly on the first try without running into some hurdles. So, to solve the accuracy problem, scientists rely on something called sequence coverage. This is just the number of times a specific genomic region is sequenced. Imagine trying to read a very faded, smudged sentence in an ancient book. If you just glance at it once, you might guess the wrong word. But if you and 10 of your friends all read it and compare notes, you'll be much more confident in what it actually says. In genomics, stacking sequence reads over a specific region essentially does the exact same thing. Higher coverage drastically improves our confidence when we're trying to identify those tiny variant typos. And this brilliantly illustrates the strategic choices researchers have to make in the lab every single day. On one hand, we have genomic sequencing, things like whole genome or whole exome sequencing, which involves reading comprehensive base pair sequences. It's a very broad, sweeping look at the data. On the other hand, we have genotyping, like targeted gene panels or SNP microarrays. Now, genotyping doesn't bother reading the whole book. It just flips to a specific page to test for specific already known variants. So every time a researcher sets up a test, they are balancing trade-offs. They literally have to weigh the financial cost, the breadth of the data they want, and the sequence depth or coverage they need to meet their specific clinical goals. Now check this out, because this is really interesting. Less than 2%. That's an incredibly small number, right? But here's the thing. If you want to identify protein coding variants, which happen to be responsible for a ton of genetic diseases, you don't necessarily need to sequence the entire genome. Whole exome sequencing, or WES, targets just the portion of the genome that actually encodes proteins. And that portion is incredibly small, literally less than 2% of the whole genome. By limiting the sequenced region to just what's strictly necessary, scientists can dedicate all their resources to stacking more reads and longer reads in that tiny 2% area. It's a real game changer. It's just a brilliant trick to massively increase sequence coverage and detection quality exactly where it matters most, without taking on the immense cost of reading the entire book. All right, let's shift gears to Section 3, Cutting and Rewriting DNA. So we figured out how to accurately read the manual, and we know how to find the typos. Now, we transition from being passive observers of our genetic code to being active editors. But to rewrite the genome, you first have to open it up. And the crucial concept here is understanding double-strand breaks. These are very precise cuts made at target sites in the DNA sequence. This targeted break is absolutely crucial. It's the essential first step that kicks off all genome editing. Because, you can't just slip a new instruction into the manual without first cutting the page to make room for it. But making that cut is really only half the battle. How do we actually write the new sequence in? Well, scientists do something incredibly clever here. They hijack the cell's own natural emergency response system. See, when a double-strand break occurs, cellular repair pathways immediately rush in to fix the cut. It's an automatic response. Scientists use this exact window of opportunity. While the cell's busy frantically repairing the DNA using its native mechanisms, scientists can step in and introduce specific sequence modifications, insert entirely new code, or even just let the cell delete a faulty sequence. We're essentially tricking the biology into doing the heavy lifting of the editing process for us. Which brings us nicely to Section 4, CRISPR and editing tools. Let's look at the actual molecular pens and scissors we're using to make those cuts. Now, you've likely heard the acronym before, but let's break down how CRISPR-Cas9 actually functions in the real world. Right now, it is the primary, most flexible, and definitely most prominent approach for precise genome editing, and it relies on two main components. First, it uses what's called a guide RNA. Think of this guide RNA like a microscopic GPS system. You program it with the exact genetic sequence you want to find, and it navigates through those billions of letters of DNA to find that exact spot. Attached to that GPS is the Cas9 enzyme, and Cas9 acts as the molecular scissors. Once the guide RNA finds the precise location, Cas9 steps in and creates that double-strand break we just talked about. It is elegant, it's highly programmable, and it's completely revolutionary. Though, actually, a quick side note, it is worth pointing out that CRISPR didn't just appear out of nowhere. Before CRISPR became the undisputed gold standard, researchers developed older, alternative engineered nucleases like ZFNs and TALENs. Now, these tools function on the exact same core concept, utilizing a target-specific cutting mechanism to create double-strand breaks. So they really paved the way for modern gene editing, even if CRISPR happens to be much faster and way more flexible today. Okay, final section. Section 5, the future of precision medicine. Let's pull back from the microscopic world for a minute and look at the big picture. Why does this incredibly complex editing matter for human health on a macroscopic scale? Well, we're basically looking at two profound applications here. Under therapeutic outlook, we have the immense potential of correcting pathogenic variants directly in a living patient's cells. Imagine treating a patient with a severe genetic disorder not by just, managing their symptoms, but by literally going into their cells and correcting the exact typo that's causing the disease in the first place. Incredible, right? But then, under reproductive interventions, we have a potentially even more profound application: germline or embryonic editing. This means editing the DNA of an embryo so that a hereditary, generational disease is not just treated, but completely prevented from ever developing in that person or even in their future children. But of course, we absolutely have to balance this incredible excitement with the current reality. Widespread clinical adoption still faces some very serious technical and clinical barriers. First up are off-target editing risks. What happens if our CRISPR GPS makes a tiny mistake and the Cas9 scissors cut the wrong part of the genome? That could inadvertently cause brand new diseases, including cancer. Second is delivery efficiency. It's incredibly hard to get these microscopic editing tools into these specific target tissues inside a living, breathing human body. And lastly, there are massive safety and ethical considerations, particularly when it comes to that germline editing and permanently altering the human gene pool. These are serious, heavy hurdles that must be addressed impartially and carefully before these tools see widespread, everyday use. Which leaves us with this final thought. We've cracked open the genomic manual, we've learned to read the letters, and we've fashioned the pens to literally rewrite the code of life itself. The science is accelerating at a breathtaking pace right now, but you really have to wonder, as our ability to read and rewrite our own genetic manual accelerates, will our wisdom in using these tools actually keep pace with the technology? It's a massive question, and it's one that scientists, ethicists, and honestly, all of us will have to navigate together in the coming decades. Keep exploring, keep questioning, and I highly encourage you to look deeper into the sources we've unpacked today. Thanks for joining me on this explainer.